Thank you everybody so much for coming, and I'd like to welcome you too, and a particularly special welcome to anyone watching online. I won't forget about you. So if you have any particular questions that come up for the question breaks, you can put them on the Science in the News Twitter feed, and we'll try our best to get to them. So good evening and welcome, and I'm looking forward to taking you today through a particular journey that I've spent quite a bit of time going through. See, about the time that I was born, my older brother was 2 years old. And if you look at videos, home videos at that time of him, you might notice that he actually very rarely makes contact with the camera, which is a little bit atypical of children who are 2 years old. And 2 years later, when my twin brother and I were kind of first starting to put phrases together and full sentences together, he really hadn't yet started to speak. And you know, when he was two and a half years old, he was diagnosed with autism. And when I was little, I didn't know what this meant, right? He was just my older brother. He was just him. And so I kind of went across the rest of, you just kind of imagine, you just don't really think anything of it. But you start noticing that your older brother's a little bit different than other people's older brothers. And first, I maybe heard the word autism, I don't know, maybe when I was 3 or 4, kind of whispered. It wasn't really well. It was at the time in the mid-90s, so still not a hugely, it was still not hugely understood, not hugely diagnosed. And it's interesting because it's been about 20 years since then, and now you know, I've spent time as a family member, I've spent time as a coach, I've spent time as a medical assistant, and now I've spent time, a lot of time as a scientific researcher, trying to understand exactly what autism is and what it really means for someone who wants to study this and wants to help kids who've got problems with their social behaviors and problems beyond that. And so tonight I'm going to take you on a little bit of that personal journey and talk to you about mostly genetics, and mostly the genetics of that using the modern tools of genetics that we've discovered and what these things have taught us about autism. So it's interesting because I'm sure everyone in this room has either met someone with autism or is related to someone with autism. And when I'm sure you can conjure a picture of that person, or maybe you can conjure a picture of a movie portrayal, kind of like Dustin Hoffman's Rain Man or some other book, but it's actually a rather difficult thing to explain what exactly autism is. And before I get into formal definitions and things like that, I'm actually quite curious to hear what people here, how they would describe it if you had to try to encapsulate autism and what you believe autism is. So I'll take whatever, I'm just curious to hear. Please.
Very sensitive to very specific external stimuli. Things like tags in their shirts or strange noises or whatever that can really get them really agitated.
Yeah, so what she was saying is that children can be very sensitive to external stimuli, things like tags on their clothes, particular types of fabrics, sounds, lights, right? And this is something that's pretty commonly seen. Anything else? Oh, don't be shy. I'm going to tell you what I think after this. Please.
I always say once you met someone with autism, you met one person with autism.
Yes, that's actually one of the first adages you learn when you work with any child with autism. Yeah, so it's hugely heterogeneous, right? It's kind of this, it's you know, maybe some of this is people are being shy, but some of it must also be, it's actually kind of hard to encapsulate what autism is. And yet we all kind of have a little bit of a gestalt, like this idea of, I can imagine what autism is, but I have a hard time describing it. Well, it was the child psychiatrist Leo Kanner who gave us the first formal description of autism in 1943. And he took a look at 11 children at the time, and he gave a description that has held up somewhat well, because when I start taking a look at all the vast heterogeneity in autism, and I say, what's common between all of these kids? We kind of come up with two major domains in the most recent definition, the DSM-5 definition. And you know, if you're wondering how these definitions emerge, they emerge by a committee of child psychiatrists who get into a conference room at some hotel and argue for a week, and then come out at the end of the week and say, this is the new criteria, this is the new description, and it's better than last time. And so, you know, when we look at this, they say social deficits and repetitive restrictive behavior, right? This is kind of the two big domains. Now, it's interesting because when you think, when you've worked with enough children with autism, you realize that some of them have a lot more than that going on too. And what you think about is that autism can actually occur in the context of broader medical conditions. So things like where someone will have physical abnormalities or heart abnormalities or any other sort of problems. And we call these autism syndromes, right? So it's kind of like the autism plus some other sort of problem. And this is about the best type of distinction we can make, right? Either autism or an autism syndrome. And just want to point out right at the beginning that we've done this by observation, right? We've just done this by looking at children, scratching our chins a little, and saying, okay, this is autism. And when you're a researcher and you want to think about the biology of these things, and you want to think about what causes these things, that makes that really complicated. So, you're going to hear me say things like, this looks like autism, a lot in the rest of this talk. And that's because that's how we define it. When something looks like autism, we call it autism. And what I want to encourage you to do in the beginning of this talk, and then I'm going to try my best to prove this wrong, is try to put autism in a big bucket. Autism, neurotypical. And hopefully by the end of this, you'll see how horrible of a construction that is, and think a little bit about why that type of definition might actually hold us back from understanding the nature and the biology of what autism does. Okay. So I've already mentioned that this is a talk on neurogenetics, or the science of using genetics to explore neuroscience and the brain. And the big question to start with is, what does genetics have anything to do with this? Is autism a genetic condition? If it is, then why haven't we found the gene? So how do you prove that something is genetic? How do you look at something and say, well, this trait is genetic, right? How tall you are, or what color your skin is, or how intelligent you are? How can you prove that something is genetic? And when there's some clues in autism that got researchers, very, very early on interested in thinking that, there's probably something really important about people's genetics that leads to their autism risk or them developing autism. And, there are clues like it shows up about 4 times more commonly in boys than girls. And it also happens to run in families a lot, right? And it's interesting, if you have an older sibling with autism, like I do, then your risk of developing autism yourself is about 1 in 5. Whereas if you just take a child and pick a child randomly, their risk of having autism is about 1 in 100 or 1 in 110. So there are some clues, but how do we prove this? And the nice thing is that nature actually gives us a really interesting experiment in the form of twins, as you saw in the previous slide. And I want you to consider a case of identical twins to start, okay? And identical twins, by definition, share 100% of their DNA. They come from the same fertilized egg that splits and develops into 2 people. And so if a trait is 100% controlled by genetics, this is a genetic trait entirely, then that means identical twins who share 100% of their DNA should always share that trait. There should be 100% concordance between identical twins. AKA, if autism is 100% genetic, then if I'm an identical twin and I have autism, then my twin is also going to have autism. Okay? And you might say, okay, well, what about the environment? That can modulate things. In this type of experiment, we kind of generally assume that twins are raised in pretty similar environments, which is generally the case unless they were separated at birth. And now let's consider fraternal twins. And fraternal twins share about 50% of their DNA. So again, if a trait is 100% controlled by genetics, it's like autism, for example, then you can look at fraternal twins and say, all right, if I have autism, then there's a 50% chance that my twin has autism. So here's a picture of me and my twin brother. We're fraternal twins. That picture, we kind of look pretty similar, but we're actually quite different from each other. And we're fraternal twins, and neither of us are autistic. My older brother is. And when you look at the data and compare twins, what you end up seeing is that there's actually a very high concordance of twins. If one has autism, then the other one does. And identical twins, somewhere between 70 and 90%, depending on which studies you look at. And for fraternal twins, somewhere between 10 and 30%. So this actually not entirely passes the test, but shows us that genetics has a really important role to play in whether or not you're going to develop autism. So, that being said, let's just take a peek of where we've been so far. We define autism clinically. What you do is you have a child having some developmental delay. They go to the pediatrician. Pediatrician says, this looks a little more than normal, I don't really know, why don't I get you to a specialist? Go to a child psychologist, a child psychiatrist, and they go and they sit down with you for a couple hours, do a bunch of checklists, and then determine whether or not you meet criteria. And there's really the autism that we talked about. There's kind of this simple autism that really represents this autistic behaviors, and then more complex autism that occurs in the context of these syndromes. And in some of these syndromes, you might say, well, maybe it's just because there's kind of horrible brain malformation and then they have autistic behaviors. Nevertheless, that still gets lumped in with the autism bucket. And finally, I've hopefully made a case so far that genetics is actually going to be really, really important when we think about autism risk. So that brings us to tonight's talk, entitled The Befuddling Brain, Neurogenetics and the Mysteries of Autism. And the goal is, I'm going to take you, even if you've never studied any genetics before, I'm going to take you through in the first part of this talk all the genetics that you need to know. And after that, we're going to get into the more fun stuff. We're going to get into the stuff about what happens when I sequence the DNA of autistic patients. And I compare the DNA sequences in autistic patients to neurotypicals. And what sorts of patterns emerge. And then once we look at that and say, what are these patterns, then I'm going to take you to my favorite part of the talk, which will be the third part, where we focus on what that actually tells us about the biology of autism and tells us about the biology of brain development. So I should point out at first that I'm barely going to scratch the surface, and I'm sure that by the time I'm done giving this talk, it will be outdated, because this is a very rapidly moving field. And I should also thank my mentor here, Dr. Chris Walsh, for helping me put this talk together. So, if I were to go into your cells, every single one of them, with very, very few exceptions, I could pull them out and put them under a microscope and look at the nucleus and look for the DNA inside the cell. And in every single one of your cells, I'm going to find 46 chromosomes. So you've probably heard of chromosomes before. There's these 46 pieces of DNA in each one of your cells. And they're actually organized. There's two sets of 23. So you have, 2 of chromosome 1 and 2 of chromosome 2 all the way down. There's a special case for the sex chromosomes, whether you are female or male. And this is standard. This is human. We have this. And if I zoom in further in each one of these chromosomes, what I realize is that they are actually DNA folded up, right? So the DNA wraps up and wraps up, helps get folded, and wraps up and wraps up and wraps up tighter until you get the giant chromosomes. So there's a lot of DNA in each chromosome. In fact, when the Human Genome Project was done, the goal was to read from the top of chromosome 1 to the bottom of the X chromosome and then the Y chromosome, and read all the sequence of the DNA. So you might have heard A, T, G, C, these things that make up DNA, the sequence that's going to be really important. And when they sequence the DNA, this is kind of what they come up with. From 1 all the way to 3 billion, which is how many A, T's, G's, and C's there are in the human genome, of sequence right here. Yay, we've done it. We've sequenced the human genome. What do we learn? Well, I know there's a lot of A's, T's, G's, and C's, but does this have any meaning? What does this tell us? So, and where are genes? We always hear about, you read magazine covers and they say the intelligence gene, the criminal gene, the gay gene, this gene, that gene. Where are they? So I want to give you an analogy here, and maybe you've seen this before, maybe you have children. It looks something like this. And maybe you've seen this, and here we have a series of letters that really just don't have a lot of meaning. All they are is just letters on a page. We can go through and read them and read them and read them. But what happens is if I look very, very carefully, every once in a while, I'll notice that the arrangement of the letters actually carries some sort of meaning. There's something there. And similarly, we can look at those A's, T's, G's, and C's in the genome, and we can determine that there are regions of the genome that carry particular meaning. All right? And we call those genes. Now, there are other regions of the genome that have meaning that we don't call genes. There's an active fields of research right now. But really what a gene is a region of the genome that carries information to make proteins. So building blocks in our body that do all sorts of things. So there are proteins that make up your muscles, and there are proteins that are important for helping you see, and there are proteins that are important for absorbing things in the gut and signaling. Everything in your body, all the machine workhorses are done by proteins. And we have about 20,000 of these regions, somewhere within that 3 billion A's, T's, G's, and C's. And now, when we look at this and we say, all right, the sequence, the actual sequence of these letters carries information for these proteins. What happens when I change that sequence? And every one of us we have very similar genomes, but there are differences between me and you and the person sitting next to you, unless you happen to be an identical twin. Now, imagine for a moment we're within a gene where there's a protein, that this blue spiky protein is what's being made, and pick whatever favorite protein you want, and you change a letter. So right now the sequence is ATCA right in the middle, and what I do is I change that C to an A. Okay, so I've just changed one base pair. That's all I've done. Sometimes what happens is that I've broken blue spiky protein. I don't get the same protein that I had before, and maybe it's not going to be able to do its job. Or maybe this same change will actually make it so I don't get blue spiky protein at all. Maybe I didn't break the actual sequence of the protein, but I broke something that's near the gene that's really important to make the protein altogether. Or there are many ways this can happen. Or, and this happens very frequently, I change the sequence and blue spiky protein is fine. There's no difference. And we call these silent changes, right? So, there's lots of ways that you can change the sequence, but you can't necessarily predict the effect. You can sometimes, depending on where the sequence changes and what the sequence changes, but the way that this variation works, you can't necessarily predict that a change in the DNA sequence is going to mean something physically for somebody. And this is going to come back, because right now I feel like the way that genetics is taught, it's often like you change this and you get this automatic change in the physical appearance of somebody. It's a little bit more complicated. So what about other types of variation? There's not just these single base pair changes between our genomes. Let's go back to the chromosomes. So imagine for a moment this is chromosome 1, right? We're right at the top, chromosome 1, and I've labeled a few regions, region A, region B, region C, region D. And each of these regions actually represent a pretty large chunk of DNA. And every time a cell divides, the DNA has to replicate. But remember, there's 3 billion A's, T's, G's, and C's. There's 23 chromosomes that each need to replicate. So sometimes it doesn't go so well, and mistakes are made. And you can get a deletion of a particular region. A big chunk right there. And this could be a big chunk on the order of thousands or maybe millions of letters. Or maybe it's not so big. Maybe it's just 10 or 20 or 30. But the other side of this is that you can get duplications. Regions that you had one C before, and now I've got 2. And we call these things copy number variants. And we all have them. They all happen. In every single one of us, there's copy number variants. Maybe not very many, but this is part of the natural genetic variation, just like sequence changes are. And finally, I just want to introduce one more genetic concept, which is the idea of de novo variation. One of the most common things that you'll hear is, well, if autism is genetic, why is it that non-autistic people give birth to autistic people? You should inherit something from your parents, right? You should inherit the autism. And while what I'll show you is that there actually is a significant amount of genetic risk that you may well inherit from your parents, a good chunk of what causes of autism-associated genes and mutations in autism-associated genes are what are known as de novo variation. So let's start with this. I'm going to show you something called a pedigree. And the idea here is that squares represent men and circles represent women. And these are chromosomes coming from the square, chromosomes coming from the circle. And when a square and a circle love each other very much, they have children. And you notice that you had 23 chromosomes, right? 23 types of chromosomes, but 2 copies of each. How that happens is you get 1 copy of chromosome 1 from your dad, 1 copy of chromosome 1 from your mom. And this happens for every one of the chromosomes, so you end up getting a full complement of 46, okay? And again, DNA replication doesn't always go right. It's a complicated process. 3 billion letters, huge organization that has to happen. And sometimes what happens in the cells that divide very rapidly, like sperm cells, and cells that had to divide rapidly in utero, like egg cells, make mistakes. And so you end up with a mutation. And so even though this type of change, whether it's a sequence change or a copy number variant or any other type of mutation, whether that happens here, it didn't happen, it's not affecting mom, but it will affect the child. And so the child may end up with a particular disease if that mutation is associated with disease. Okay, so we're covering about 2 semesters worth of college genetics at a very rapid rate. And the basic things I want you to be able to take away from this is that the sequence of the letters in DNA is actually pretty important, carries meaning. And we all have a lot of variation naturally within our genomes. And so what we can do is look at the types of variation that exists and see what types of variation are associated with disease. And that can start our hunt towards understanding the biology of something. All right, so I understand I just went through a lot of genetics, and I'm just curious if anybody's got any questions. I can answer them right now. And if not, we'll move on, but please. Any questions? Doesn't have to be about autism, that genetics is fine. Right, no.
I'm not sure if you're going to cover it, but
Yeah.
Is there any percentage correlation between what percentage of autistic people are savants or have some prolific or profound skill in some?
Yeah, so I won't talk about it specifically, but yeah, I'll let me address this, because this is another one of those things you think about when you think about autism is savant skills, right? Maybe I'm kind of, I've got a lot of deficits in one area, but I'm a musical genius or a mathematical genius. And these are things that are very commonly thought of as part of the autism picture, but are actually relatively uncommon. Very, very few autistic children have savant abilities. But when they do exist, and is there a difference between the kids that have savant abilities versus kids that don't? You know, these are the types of questions that are very interesting to examine, but it's kind of hard to get that type of research study to work. But that's something that people are looking at and do look at. Please.
So in the lab,
Yeah.
not a scientist at all, I'm a teacher, but in the lab, and you're like cutting up these genes,
Yeah.
changing the A's for the C's. Like, how does that work? And how do you know that a protein is changing?
Yeah.
What are you observing?
That's a really, really great question. So we start, and so let me repeat that question, is how do we know that when we change letters and that we get changes in protein and function? Like, there's a lot of how do you know? You can't see DNA, right? Like, it's kind of hard to look at. And the answer is that there's a lot of different tools that have been developed over decades and decades that are used to manipulate DNA. And we can actually sequence DNA pretty well. So if I have a test tube with a cell in it, I can send that, or you need more than one usually. There are technologies that work with one, but if I can send that to get sequenced, and I can get the genome sequence in order. And if I want to change sequences, there are different tools for doing that. Mutagenesis, or you might have heard of this fancy new thing called CRISPR. There are ways you can change sequences. Proteins are a little bit more complicated, right? Because every protein is different. So maybe I think this protein is really important for anchoring a cell to something. And what happens when I change the sequence is that cell no longer anchors well and it floats. So it's a functional readout. Or I can actually look for the specific protein itself with an antibody. And I can look and say, is that protein where it should be inside the cell? If it's on the membrane, where it's in the nucleus, is it there as much as there should be? So there are lots of different tools that biological researchers use for this.
So it's just how it affects that one or two cells that you're observing.
Yeah, so that's on the cell level, and then we can also do bigger on the organism level, right? So I can change a sequence in a mouse, for example, and then maybe that mouse doesn't look like a normal mouse anymore. Or maybe it looks exactly like a normal mouse, and I don't know what I just did. And you know, there are ways that you can, and I used to do mouse work, so. So lots of different, there's a lot of tools that have been developed over a long period of time that help us understand. People use mice, they use flies, they use worms, and they also look at like disease patients, for example, who come in, like rare patients who have mutations, and you can see what sort of clinical syndrome they have, and see if they've got a mutation in a gene that you think might be associated or might be causing their disease, right? You'll notice that I'm straying very, very carefully from saying like, definitely this gene causes this disease, or this gene causes this phenotype or physical characteristic, right? The classical way we're taught genetics is with Mendel and Mendel's P's, right? And they say dominant and recessive and things. Those are useful terms to some extent, but they really represent very specific cases where a particular change in a protein has a big effect on a physical outcome, and very particular changes have those big effects. Most of genetics doesn't work like that.
Is there any correlation for de novo variation and environmental exposure, like during embryonic development?
I don't know. That is a great question. The question is there any correlation between environmental exposure and de novo mutation, right? Do people who have particular environmental exposures have a higher rate of de novo mutation? I don't actually know the answer to that, but we will talk a little bit about de novo variation, where it comes from, and why it seems to be important in autism, and what that tells us. That's a great question, though. There are things that can affect development in utero if you take certain medications in pregnancy exposed to certain toxins, but how it affects mutation rates in DNA, that's a good one.
Yeah. There's a lot of
expression of something will change. The actual DNA sequence may not necessarily change, but a really good example is the Dutch hunger winter that happened, I don't remember when, it definitely happened. Women who were pregnant at the time had really, really low nutrient intake, and their children ended up having very, very slow metabolisms when they were older because their bodies had already been trained in utero to kind of retain calories and store fat and really avoid expending too much energy.
And development is a very dynamic process that involves input from genetics and environment. And also, depending on your genetics, you may respond to that environment differently, right? So this is something that, to look at something is like caused by particular sequence changes. That's why I'm trying to be careful with it. But that's a fantastic question. Okay, so now let's take a look at what happens when I go through with modern tools and I say I want 10,000 children who are neurotypical and 10,000 children who are autistic, and I want to sequence their DNA and just compare and see what sorts of differences I have. Can I explain somebody's disease by sequencing their DNA? Right? This is something that genetics gets sold on a lot. We can unlock the human genome and explain everything, and now we're going to have a great understanding of what's going on if we can just read those letters. And let's go back a little bit to that distinction that we talked about with autism spectrum disorders, kind of the big picture, the big bucket. And then there's the autism syndromes that are in the middle, these more complicated autism plus, right? And early on, when genetics was a much earlier stage, before we had sequenced the human genome and we could really only work with particular genes, particular areas, researchers wondered if some types of autism, right? We look at particular clusters within autism. These kids look kind of similar to each other and they all have autism. They all have autism, but they also all have craniofacial abnormalities or all have cardiac abnormalities on top of that. So there's a picture here. Is that cohort, can we explain that with a particular type of mutation that's within the bigger picture of autism? And so these autism syndromes, what happens is they kind of fall under the more classical model of looking at genes and disease, where you end up sequencing people or you've got a particular gene in mind and you find that it shows up again and again, and that anytime you've got a change in that gene, you end up with a particular type of autism syndrome. And there are several of them. And the first one that was ever identified was a disease called Fragile X syndrome. And most kids who have the mutation in Fragile X syndrome that is pathogenic or a problematic mutation end up with autism, right? They look autistic. And they have other sorts of issues too, but this is like about 1% of children with autism in general actually have Fragile X mutation. And there are other ones, right? There's one studied by another one of my mentors here, Dr. Mustafa Sahin, who studies a disease called tuberous sclerosis. And more than 50% of children who have mutations in this TSC1 or TSC2 gene are autistic. But we wouldn't necessarily have known that before we sequenced them. They just kind of all fit in that clinical bucket of autism. And when I look and look at all of these autism syndromes, and let's say for a moment I had 100 autistic children, okay, that make up this pie, and I want to explain all of their autism. If I go through and I look for autism syndromes, these syndromes where we have autism but this other side of it, I'll explain about 5 of those children. I'm going to say, okay, I'm very confident now that the thing that I've just identified is causing this person's autism, and there are other problems. And these tend to be single gene disorders, okay? One gene, you break something, it breaks a protein that does something pretty important. FMRP does a lot of important things. We'll get into a little bit of that. And the other side, this one more kid I can explain with some really severe chromosomal problem, right? So a big deletion or a big duplication, something that's huge and clearly very disruptive. It might disrupt hundreds of genes. Okay. So that's 5 or 6 kids out of 100 that I've done with this kind of old school method of thinking about autism syndromes. What about the rest? What about the other variation that we talked about? If I compare variation in sequence changes, and if I compare variation in copy number variants, might I be able to explain pathogenesis of autism? And so let's go back to that copy number variance, and remember that many copy number variants are very common. You can have deletions, duplications, they can be anywhere in the genome. They can be big, they can be small, but they're a particular type of change in your DNA. And what happens is when you go through, and let's design an experiment really quickly. I'm going to have a family, remember, square circle, mom and dad, and I'm going to have an autistic child and an unaffected sibling, okay? So you think that they share the same genetic background. So things that risk that you might inherit from your parents, that's pretty similar between the two. And yet one child developed autism and the other one didn't. And if I go through and I sequence genomes comparing these two, and I look for copy number variants, what I find is that the autistic siblings actually have a higher burden of rare copy number variants. That's kind of important. Not common copy number variants that everybody has, but rare copy number variants. And when we look at this, you see there's in pretty equal proportions of deletions and duplications. So it's interesting because they have the same amount of common copy number variants, but they have these ones that don't show up in everybody. They only show up in people with autism. And this explains maybe another 10 kids in that 100. It's copy number variations that are disrupting some sort of genes somewhere. Okay? Disrupting regulation of those genes, disrupting those genes, but it's big chunks of sequence changes. What about those individual sequence changes? 1 base pair, 2 base pairs, right here and there. And ones that are particularly important, not the ones that break proteins, meaning that I don't get a protein produced or I get a completely non-functional protein. So I go through, use the same experiment, same genetic background, but I'm comparing an affected child to an unaffected sibling. And I compare, well, it turns out that the autistic population actually has a higher burden of damaging point mutations or damaging sequence changes, but not silent ones. So those mutations that don't change protein function, we all have them. Turns out that autistic children and adults actually have more of the damaging kind. So if you were to say, hey Sham, maybe this is just a disease of mutation. Maybe there's something that causes these children to have more mutations than other people. And I'd say probably not, because they actually carry the same amount of sequence changes in general, but they tend to have more of the bad kinds. So maybe this looks a little bit like you just get unlucky on where that mutation occurs. It's a natural part of cell division and the amount of cell divisions that you need and the 3 billion base pairs and making trillions and trillions of cells. Sure, errors are going to happen somewhere. So these point mutations, they're another 15%. So now I'm up to about 35%. Most of these are de novo processes. Don't happen in the parents. I can sequence the child, sequence the parents. The child will have the change in the DNA sequence, but the parent in the same place in the genome doesn't have a change. So we can say that this mutation occurred de novo in the child. So I can go through and I can look at other types of mutations, things that we haven't necessarily talked about. Rare inherited variants. You find these by looking in families with consanguineous marriages. You generally have to go to other parts of the world where rates of those types of marriages are higher, and you can tend to find, it kind of gives you a little bit more statistical power to detect variation that might be rare inherited variants in autism. And there are other types of things that are really currently being explored right now. Right, so I mentioned before this idea of background autism risk. And we think that a lot of that risk is actually carried in common variation, not just variation that is rare and that we know is damaging, but variation that we all carry, right? So that's probably some of this. We think that there are things called somatic mutations, meaning mutations that change just some of your cells. So imagine I'm an egg, a fertilized egg, and I'm going to turn into a human being. I go from 1 cell to trillions. And maybe the mistake in replication doesn't happen in the fertilized egg, in the sperm cell and the egg cell, but it happens in round 2 of division when there's only 4 cells. And now 1/4 of the cells in the person from now on will have that mutation, right? So maybe this has something important to do with you get a milder autism, you get a milder disease, or maybe it explains to us why when I sequence people and I take skin samples or spit samples, I don't find mutations that I would expect. Maybe it's only in some of their cells, right? These mosaics. So, this is a big chunk of the pie that we're trying to explain, the autism heritability. Heritability meaning the amount of this that we kind of attribute to genetics. There's more formal definitions of heritability. I can feel my professor like somewhere feeling a disturbance in the force, feeling like I just gave a horrible description of that. But think of this pie as the amount of autism that we can kind of attribute to genetics. Okay, so I spent this whole time up to now telling you about single gene disruption, sequence changes, copy number variants, all of this variation. And I'm saying it's disrupting genes. And okay, now let's see, what genes are we disrupting and what do they do? Well, I told you about FMRP, it's on the X chromosome over here, and we disrupt FMRP and we get Fragile X syndrome. And well, there's TSC1, that's the other one I mentioned earlier. And what about those copy number variants? Well, there's one over here, 16p11.2. It's the most commonly associated copy number variant with autism. And there's another really interesting one on chromosome 7. So what's particularly interesting, because this is a unique copy number variant, where if you have a deletion in this region, you don't get autism. You get a disease called Williams syndrome, where it's kind of interesting because these are kids who usually have some intellectual disability, and they've got facial abnormalities, and they've got cardiac abnormalities. You shouldn't touch the mic. But the interesting thing is that they are really hypersocial, and they look at you right in the eye when you talk to them, and they want to keep talking to you the whole time you're there. So sometimes people come and look at that and they say, whoa, increased social motivation, kind of increased social function beyond what you would expect for someone's intelligence. But when I get a duplication in this region, you get autism. So maybe there's something interesting going on in 17p11.23 that has something to do with social motivation, social behavior. And that locus has 28 genes in it. Okay, which one of those 28? There's another gene that I worked on when I was an undergrad called SELF6. All right, well, we found SELF6 by sequencing autistic patients, and then you go through and we break it in a mouse and see what happens, and it turns out the mice act kind of weird. And what does it do? Well, it's an RNA binding protein enriched in serotonergic cells. Oh, great. All right, that really doesn't help us very much. But it's interesting because, when I was preparing for this, I wanted to go through and look at and see, what else is coming down the pipeline. And this is a paper from March in Nature Neuroscience, one of my favorite pastimes in reading. And they do whole genome sequencing, and they say, ooh, 18 candidate genes identified, right? That's great. And this is one from actually just a couple weeks ago, Neurogenetics, another one of my favorite pastime readings. And 91 neurodevelopmental disorder risk genes with autism and developmental disability biases, right? Again, everybody's being careful. They're not autism genes, they're associated genes. They're neurodevelopmental disorder genes. Want to be correct in these. But if I go through and I start collecting all the mutations we've talked about, all the copy number variations, all the sequence changes, that chromosome map that you saw looks something like this. Yikes. This is in 2015. At that time, they identified about 800 autism-associated genes. Some of these, you can get single gene disruptions and you get autism. Some of these I found by just seeing there's more commonly mutations in these genes in autistic people than there is in neurotypical people. Some of these are genes that exist within those copy number variants that are associated with autism. 800. And that was 2015. If you look at it right now, it's about 2,000. Yikes. I call this the who's list of autism genes. Right? There's, really cool ones like SELF6 I told you about, RB-FOX, that's another one I work on, right? But I get to pick my favorite ones and kind of explore that. Now, a lot of people just pick their favorite ones and go with it. Probably not the best strategy. So, we talked a little bit about what happens when we sequence people with autism and compare that to sequences of people who don't have autism. And what we realize is that autistic patients have a higher burden of rare copy number variants and damaging types of single sequence changes, and that these mutations are frequently de novo. All right, let's pause for a moment for some questions, then we'll take a break, and then I'm going to take you through the most fun part of this talk. Please.
So, sorry, I'm asking some questions.
No, please.
Is it that in 2015 there, I mean, you don't know the answer to this, but I'm asking you, that there were 800 of the who's who, and it's on the rise, it's increasing? People are saying autism is on the rise, or is it just that scientists are discovering more? Yeah, so I do actually have an answer to this because there's a lot of opinions on the matter. So you often hear, the question is, there were 800 genes in 2015 and now there's 2,000. Is there just, does this explain why people say autism is on the rise? So when my brother was diagnosed in 1994, he was told that about 1 in 2,000 children have autism. And now, when I get up and I start to teach about this, one of the first things you always kind of just say, because it's something that's associated, is 1 in 110. Or you'll hear numbers like 1 in 88 or 1 in 66, right? You hear this. Wow, no other disease in the world has had that kind of ridiculously rapid rise. So what's causing that? Is there something that we're doing? Is there some environmental problem? Are human genetics just mutating really crazily? Well, I want to invite you to think about what we talked about right at the beginning when I said that what we do when we define autism. Get some child psychiatrists in a room, have them argue a bit, feed them a little, and don't let them out until they say, okay, we agree. And they say, this is what autism looks like.
And what I really think, and I think the majority of people who study this, is that, remember, we call what looks like autism, autism. And what we've defined as autism maybe 50 years ago was like, well, Fred's just a little bit off. And the other thing is diagnosis is now really important for securing services in schools. It's just it's much more part of the consciousness of how people approach their children. There are higher rates of picking up developmental disorders by pediatricians. So the best guesses, if you asked, I'd say 98% of people who work in this field, is that this is really a question of diagnosis and definition rather than a question of a rapid rise in autism.
Some people say it could be a form of evolution.
Yeah, so no, we'll get back to that. We'll get back to that by the end of this. Please.
Are there any kind of hard metrics, that are being used now to, in addition to the subjective evaluation, a bunch of experts, that looks like autism, any kind of standardized testing that can be repeated as you progress longitudinally in studying these people?
So the question is, are there any hard metrics that are used in conjunction with observation that when you determine whether someone is autistic or not, or being developed right now? And I'm being a little bit unfair to my colleagues in the behavioral sciences and the child psychology when I say that they just look at a child and say you're autistic. They're actually, they use standard measurement tools that are all behavior based, right? So there are interviews like the Autism Diagnostic Observation Schedule, ADOS, for example, which is kind of like the standard one used when you're enrolling people in research. So those scales have been validated across many thousands of children, and they tend to work pretty reliably. Again, even they're not exact. They're based off of whether you check off behaviors, and you have to assess them. So you get to a point where you see so many children that you start getting a feel for whether that actually meets the criteria.
I mean, like performance on like some sort of specific task or something like that, where it's like very repeatable, and you can just say, take somebody, okay, do X, follow the dots, do whatever it is.
Yeah, so you're asking if there's any particular tasks. So these interviews actually have observed tasks that have like certain metrics of what a child should or should not be able to do at a particular age. I don't think they're quite as neat and clean as you would want them to be, and they're certainly not yet biological markers, right? Like I can't, and this may change in the next 5-10 years, I can't put a child in an fMRI scanner or an MRI scanner and say, what's going on? There's really interesting research by a group called the IBIS Network that tries to look at what they do is they have children from birth that are siblings of autistic children. So they're at much higher risk of developing autism. And so they kind of pick this cohort because they know about 1/5 of the kids will go on to develop autism. So what they're trying to do is take brain imaging do that early before the child gets a diagnosis to see is there anything that you can catch early on. So maybe in a few years you can get to a point where we can screen a little bit more carefully, or we can think a little bit more carefully if there's higher risk for the child. But right now there's no good biological markers. Go for it.
So I assume there's equal study for the high variance, people that have as much variance as the autistic children that don't have autism?
Yeah.
Are they looked at closely for other genetic disorders or other, do they fit a bill? Are they also great athletes or something? People that have all these mutations that aren't autistic?
Yeah. It's interesting because the types of mutations that the unaffected people have are actually different types of mutations. They tend to be silent mutations. They tend to be mutations that don't change protein function, but behave observationally. Do unaffected siblings, do they look a little bit different than the rest of the population? Is that where you're going with this?
The group you showed the chart where it showed that they had a much more high frequency of the damaging changes. So I'm saying, do the people that had that high frequency that didn't have autism also get studied equally for traits that could, someone that has autism might have the trait, but it might not be seen as a negative trait, or it's something that doesn't get lumped in?
So for people who have that many mutations but don't necessarily have autism, I don't know. That's a good question. I don't know. I'd be curious to see that. I don't have any. I also don't have any prediction actually for you.
That's my prediction.
Yeah.
I think I have two questions, and they're related to what you mentioned about somatic mutations. The first simpler one is, do we understand the distance in the number of DNA copies that take place for different cell lines, like your sperm versus your brain cells?
Yeah.
Are they a different distance from your original embryonic cell? So that we know how many opportunities, is there a lot known about the number of opportunities for a copy error for these different categories of cells?
Yeah, so I'll start with that question, which is, when you think about somatic mutations, you think about a fertilized egg, and that'll end up developing into a human that has brain cells and sperm cells and other types of cells. Do we know how many divisions you are away from that fertilized egg to one of those final terminally differentiated cells? And that might tell us about whether a cell has some more risk of developing a mutation because it has to divide more times. So the answer is that's actually being investigated very actively right now, and it's the kind of thing that's being investigated in my mentor's lab, Chris Walsh's lab. And it's interesting because, you know, it's not, I say that there's a lot of problems with DNA replication, but considering that you have 3 billion places to make an error, it's actually pretty amazing that there's not more. And considering that, like, when I go through this sometimes, I think about, like, I'm currently in medical school right now, and I sometimes walk around thinking, like, goodness gracious, how am I still standing? Like, there's so many ways for things to go wrong. But many times it actually works out just fine.
So we don't know yet?
No, not yet.
Okay. So I guess we won't know, but I guess my next piece of that was going to be, whether we knew that or not, do we have any understanding of which parts of you share ancestry going back how far in those cell lines, so that we know whether taking, I guess what I'm asking is, you meant, like, I think it probably wouldn't matter if my elbow has autism. Yeah. Right? And so you would want to know whether you are learning about the DNA of my brain when you sample my skin. Yeah, so that's actually exactly the kind of question that's being investigated in my mentor's lab right now. Because a lot of this, when we take samples of people's DNA, we do cheek swabs, we do skin samples, maybe blood draws. And, well, what if, in general, the embryonic patterning means that I'm more like the blood sample is very similar to the type of somatic mutations I'd have in my brain, but the spit sample is not, right? So maybe then we should be making sure that when we sign people up for research in this, that we're actually taking blood draws before we do sequencing. Or if the sperm cells are mostly not shared with the brain cells, then your autism would have less to do with your children's.
Yeah, exactly. Go ahead.
I was curious about countries like China, where there's a single child policy. We made a statistic earlier that it's, we observe it's 4 times more common in sons than daughters. So are we seeing in a country like China, where there's historically or recently been, you know, if you get your first son, you just quit and you leave it there. Is it closer to 1 in 66 versus 1 in 110 in a country like China? Do you see any of that?
So doing prevalence estimates of autism in general is hugely controversial. And the question is, do we see any change in prevalence in autism in other countries where you kind of have a single child or smaller family size than you would somewhere else? I don't know, because the prevalence estimates are difficult to measure, and there are people who argue between 66, 88, 110, and 266, whatever it is. And we know that there is cultural variation in whether or not autism gets diagnosed, right? So it's only recently in the past maybe 10 or 15 years that autism has picked up a significant amount of awareness in China. I mean, it was there before, just like it was in the US, but part of trying to make estimates like that is actually being able to diagnose and have a systematic research structure. Yeah. In the back.
I'd like to know how much the epigenetic have an effect on autism. So, as we spoke about genetics, all the other part is actually epigenetic.
Yeah. Oh, you'll notice that I didn't include any slides on that because we don't know. The question is, how much does epigenetics contribute to autism? So for anyone who hasn't heard so much about epigenetics, so we talked right now about changes in DNA sequence and the actual DNA structure. But whether or not a particular gene is turned on or off, whether the protein actually gets made, is different from cell to cell. In fact, that's part of what makes a brain cell and a skin cell, and so on and so on. So the regulation of whether genes are expressed and how they're expressed also has a role in disease. And this is something that I really hope people will start answering more. Maybe they have, and I just am not really that aware of it. But I'm trying to think if there's any epigenetic associations, and I can't right off the top of my head. So we'll come back to some of these questions at the end of the talk. And before we continue on, there was one type of question about increased risk with cell divisions. And there are some things we do know, like if the older your father is, that increases your risk for autism, because as your sperm, the cells that make your sperm age with you, the germline cells that are there will accumulate mutations, right? Just like you're more likely to have a child with Down syndrome if you're an older woman, because the eggs will start to degrade a little bit and have non-disjunction, where you get more likely to have chromosome abnormalities. So there are things that we know about mutation accumulation that increase your risk for autism. Was there one more question?
Yeah, isn't it difficult to get information about like population's genetics from other countries because they just want to keep it, well, not secret, but
yeah, these things are very difficult. And the question is it difficult to get information about genetics in other countries because it's difficult to share data and share? It is difficult, but it is being done, right? So much of the sequencing that I'm talking about has happened in the US, but there have been identified families with high rates of autism and certain types of populations that are studied in other countries by researchers in other countries and the US. And the idea is that as we move forward, our databases for genome sequences should be international, and they really should be shared across the world, because that's how we're going to make sure that we're doing a good job of sampling and we're getting a good characterization of the real genetic variability across the world. Yes.
You mentioned that being male is one of the genetic risk factors.
Yeah.
But then in your withdrawal list, it's spread all over the genome.
Yeah.
So how do you explain that males are more risky?
This is a fantastic question, because when researchers first said there's 4 to 1 males to females, you know what? This is going to be on the X chromosome. We're going to solve autism by finding mutations on the X chromosome. So males in general are more susceptible to mutations on the X chromosome because they only have 1 and females have 2, so they have another copy sometimes. Females have another copy to make up for it. But it turns out that that's not what happened. You look and it's all over the genome, right? It's not just like it's all over the place. Yeah, there are plenty of loci that are on the X chromosome, and there are autism syndromes that only show up in men, or only show up in women, or only show up in men, and that is based because of X chromosome patterns of inheritance. But how do you explain that? And the answer is that there's this is now a hypothesis. This is not definitively proven, and I'm sure there are people in my field who would disagree with me entirely, is that there's something about being a girl that is protective. And you can have very similar genetic liability being a boy or a girl. And girls are then less likely to go on to develop autism. So there's actually something specific about females and female biology that that's a protective factor. I had a mentor who used to say that, you know, what's the best defense against being autistic is being born a girl. So it's a very, this is one of those places in the field that I think once it gets broken, that'll be a big clue towards where we're headed. I'll take one more and then we'll move on. Yeah.
I just wanted a quick aside. I heard that like males have higher variance, like males and females have the same IQ average, but males have higher variance. So I wonder if it kind of all goes back to the variance.
Variation. The question is that it's thought that men have a greater variance in IQ than women, but both men and women have the same mean IQ. I'm not sure how that relates directly to this. I'm actually not sure that that's entirely, like, IQ is not my favorite measure of physical phenotype. It's actually a very weird and difficult thing that relates. Does it really relate to intelligence? How it relates to intelligence? Measuring intelligence is one of those behavioral things that usually I leave to my psychologist colleagues, but I'm not sure exactly. But when you think about overall variance in what we'll call autistic trait, which we'll get to by the end of the talk, there might be something to that, but not necessarily for IQ. Okay, so why don't we take an intermission right here, and we'll take like maybe 10 minutes, and anyone who's got specific questions, come on up to the front. I'd love to talk to you. And then we'll come back together at like 8:07, and we'll finish this talk, because it's the most fun part is yet to come. Okay, so I understand that I've taken you from what is a gene to, I'm going to take you through now functional gene networks and particular examples. So we're kind of pushing, I'm going to try to push it a little bit in terms of where it is. So a lot of this, you know, there'll be lots of time for questions at the end of it. But let's return back to the idea of this giant map of autism genes. And let's return back to this idea and say there's now, what, 2,000 of these that have been identified. And you start asking yourself, well, what the hell are all these genes doing? Like, there's so many of them, and it looks like I can break multiple different ones and still get something that looks like autism. So is there any sort of pattern? Are they doing similar things? You know, if I break something, do they converge somewhere on some node that I can look at and say this convergent pathway is really what's going on in autism? And the other side of this is that there's really no such thing as an autism-specific gene, because when I do this same type of experiment for ADHD and schizophrenia and obesity and epilepsy, I find huge overlaps. The genes that we have identified associated with intellectual disability have a gigantic overlap with genes that are associated with autism. And that kind of makes sense, right? I told you that many, many children with autism also have a degree of intellectual disability. And many, many children with autism develop seizures. So it makes sense that there's an overlap. So maybe this overlap can be instructive in what these genes do. But again, no such thing as autism genes, autism-associated genes, and nervous system-associated genes. So it's not just about what the genes are. It's also about what type of mutations you have in them. It's also about what types of cells have those mutations. And now, I'm going to pop up this figure over here. So we have this pattern of 2,000, 2,500 autism genes or so. And we're trying to look for patterns. And what researchers did, this is a Krishnanan Nature paper, 2016. And they took this, they put really big computer algorithms to it and said, I'm going to see what every one of these who's lists is associated with, what types of functions they're associated with, and see if there's any sort of pattern. And this is a little bit arbitrary, because part of this is someone looking at a gene and looking at the literature associated with that gene, the research that's been done, and saying, well, this gene is associated with synaptic transmission and plasticity, things associated with the brain. So I'm going to tag all of those genes associated with that in red. And when they do this, though, and they say, is there any sort of pattern? By any way I cut this pie, can I find patterns? They find patterns. They find these associations. And maybe these clusters of autism genes, when I put them in a big network map, can tell us a little bit about the biology of autism. What actually happens when I disrupt these genes? Because remember, it's not gene to disorder, right? Gene to protein, to function, to cell, to circuit in the brain, the cells talking to each other, to organism. There's a lot of layers between a change in a DNA sequence and a physical disease. Okay, so we've looked at this we're looking at this big functional map of autism genes. And we say that there's a big cluster that seems to be associated with synaptic transmission and plasticity, right? All of you here have just come for a talk about the befuddling brain, and we're finally getting to the brain part of this talk, which as someone who was trained as a neuroscientist who's been sucked into the world of genetics, it's a good relief for me too. So what synaptic transmission and plasticity means is that we're now thinking about how neurons in our brain talk to each other, okay? There's a lot of neurons that make up your brain and nerves all over your body, and they have to communicate to each other with signals in order for us to think and do anything that we do, me moving, speaking, all of those things. Now, when we look at this particular cluster, and we're just going to put a picture of a synapse, okay? So maybe you've heard of a synapse before. This is the space between nerve cells. This is how nerve cells talk to each other. So we've got a cell that's talking to this cell. So this is the end of one cell, the beginning of another. And the synapse is a space between the ends of those cells. And the synapse is a really important part of the cell. It has a lot of organization. And there's a lot of very particular proteins that make a synapse function. So is this cell going to be an excitatory cell? When this cell fires, does this one get excited too? An inhibitory cell? When this one fires, it quiets this one down. And depending on how the relationship of these cells talking to each other, the structure of the synapse looks different, which means the composition of proteins at the synapse looks different. And when we look at that who's list of autism genes, we actually find, like the cluster shows, that a lot of them are in the synapse. A lot of these proteins function in the synapse. So all 4 of these are on that giant list that I showed you, and most of them have been associated with syndromic forms of autism. So kind of the more complicated ones that cause these single gene disorders. Most of these that are up here. And I want to focus on one right now called SHANK3. this gene has gotten a lot of attention because it has an overlap, just like all the other autism genes do. It has an overlap with schizophrenia. And recently, researchers went through and they looked at particular types of mutations and were able to show that depending on a type of mutation you have in SHANK3, that'll raise your risk for autism or raise your risk for schizophrenia, right? And so the difference in mutation actually has a functional consequence. But this is an important protein for the synapse. So now let's look at an electron micrograph of a synapse that was pulled from a mouse. And here's the cartoon. So this is the presynaptic cell, the cell at the top. Notice that there's these little circles here. These are vesicles that contain neurotransmitters. So that's how the cells communicate across the synapse. And now we've got this postsynaptic cell over here, and they've labeled it in purple, okay? So this is the cartoon version. This is what it actually looks like. Very, very, very small space, super highly organized, okay? And now what I'm going to do is and this work was done a couple of years ago, but you take a mouse and you break SHANK3 in the mouse, okay? Just like it would be broken in particular types of autistic patients. And I'm going to take the same type of picture, and it looks something like this. Now, I know this is really subtle. It looks like, oh, that looks exactly the same. But when you look at hundreds and hundreds of these, like electron micrographs, and get really used to what the synapse should look like, this is actually pretty dramatic, right? So first of all, it's smaller, it's a little flatter, and the organization that you see on the top actually gets disrupted, or the bottom actually gets disrupted. And you can't really tell just from this image, but the idea is that when I disrupt SHANK3 in the mouse, there's a big change in the organization of the synaptic structure. And the SHANK3 mice actually show behavioral abnormalities as well. So that's pretty cool. What? But this diagram, this cartoon, is really the bigger picture of all those who's list of autism genes in the synapse. Looks something like this. And this is, again, simplified. When I say there's a lot of moving parts, I mean thousands of moving parts. And every single one that's labeled blue and purple are the ones that have been associated with autism. And sometimes they're associated with that syndromic autism, single genes causing autism symptoms. And sometimes they've been associated with non-syndromic, right? This classic non-complicated autism. And there's our friend SHANK3 and mTOR. This right here, you can't actually see the gene labeled, but that's the TSC1 and TSC2 that we talked about. FMRP, that leads to Fragile X. This leads to this. This was done by a group led by Huda Zogby. And it's really interesting because what they did is they looked at all the patterns of synaptic associated genes in autism, and they came up with this, well, here's this kind of summary picture of it. But the picture is really, really complicated. So then, is autism a disease of the synapse? Maybe there's just something different about the fine-tuning and the way the neurons communicate in somebody who's autistic compared to somebody who's not. All right, let's go back to this cluster diagram again. Another one of these clusters over here. This one isn't associated with the synapse. It's associated with something called splicing and mRNA transport. For anyone who has been to this series of lectures, one of the lectures before this by Heather Landry was on splicing. She did a fantastic job describing what is often a neglected process in biology. And splicing in mRNA, oh God, that has nothing to do with the synapse, except for synaptic proteins have to undergo splicing. But this is a big, broader category. And what are some genes that are in this category? And these are genes that I've done a little bit of work on, but are work done by Chris Walsh's lab and by a postdoc in the lab named Xiaocheng Zhang, and he was my mentor this summer. And we worked on a group of genes called the RB-Fox genes. And these are really important because they always show up on that who's list of autism, right? RB-Fox, 1, 2, there's 3 of them. They all do pretty similar things. So there's kind of a redundancy here. And they seem to be involved in splicing. When I say splicing, if you haven't heard of that, just think of it, it's important of how we get from DNA to protein, okay? So we get from the point where I have my sequence of DNA to a functional protein. Splicing is one of those steps. And if that goes wrong, you can end up with all the same types of problems with the protein that the sequence change can lead to. So splicing changes can lead to disease. And now the question, we know that it shows up in this who's list of autism, and I have to say, well, how does changing RB-Fox or getting rid of RB-Fox, breaking RB-Fox, how does that affect brain development? How does that affect, how might that lead to an autistic phenotype? So here's a cartoon of kind of normal brain development. Came out of Walsh Lab itself. And so early on, think you have neural stem cells. You hear a lot about stem cells. Stem cells just end up making lots of differentiated cells eventually. And they divide and make little baby neurons and little baby support cells that make up your brain. And your brain kind of gets formed from inside to out, okay? So divide, 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 and the cells migrate. They actually use the scaffolding of that stem cell to climb their way to where they should be. And the brain gets built from the inside out. How else would you get such exquisite organization, right? There's got to be some sort of really organized method. When you're born, you have 100 billion neurons. 100 billion. That's the same number of stars in the Milky Way galaxy. And they're pretty well organized. We can actually count the layers and see different functional organizations. So this has to be very tightly controlled. So now I'm going to show you an image, and can you see the green at the very top of this? Yes. You can see it okay? All right. So this is a picture of mouse cortex. So not quite as complicated as human brain, but they actually have very similar layering patterns. Ours are a little more complicated, but they've got the same basic structure, right? So cells would have to start at the bottom when they're forming, and as the brain develops, they're going to migrate their way to the top, and they're going to reach their top layer, this top layer. So I'm really interested in this top layer. It's kind of important for a lot of things, but mostly I'm interested because these cells have to undergo a really severe migration process, okay? And I've labeled some of these cells green, okay? And what I did, actually my postdoc did, and I helped them analyze this, is look at what happens when I break RBFOX 1, 2, and 3. I don't want any of this redundancy protecting those cells. I want to break RBFOX and see what happens. So break RBFOX 1, 2, and 3 in these green labeled cells. So I didn't do it for every neuron in the brain, just for the green labeled cells. Okay, so here's a picture of what happens when you have a mouse that you break RBFOX in a particular group of cells. It looks something like this. Kind of striking. A lot of cells get stuck at the bottom, never really make it to the top. Some of them make it up to the top. But there's something interesting going on. When I've broken RBFOX, I've broken something that helps other proteins go from DNA to functional protein. But RBFOX doesn't have one target. It has many targets that it helps splice. So something about the targets of RBFOX are going to be pretty important for neuronal migration. And something about the group of genes that are regulated by RBFOX, and the group of proteins that are regulated by RBFOX, may be important in telling us about the biology of autism. Okay, so we're not going to go through every one of these little clusters because we'd be here all night, and frankly I don't understand enough about this to really go through all of them. But there's some big categories, right? This category of synaptic function or embryonic development, ooh, that seems like it might be associated with brain development, cell signaling, brain machinery, right? These are things that people are looking at. There's these patterns within the autism-associated genes, and these are the convergences. So maybe, does this have anything to do with when we think about defining the disease, right? So instead of going from gene to disorder, if we start connecting the dots a little bit differently, just like I mentioned before, it's a little bit of a complicated route. Gene to cell type affected, to the circuit in the brain, to the symptom that's actually shown up when someone walks into the clinic, to the disorder that we call them. Maybe we can be a little bit more precise about that. And what about treatment? Does this clustering pattern tell us anything about the types of treatments we can use? So I mentioned my brother early at the beginning of this talk. He's diagnosed at the age of 2. Generally, the earlier you're diagnosed, that's not a good sign because it means you've got pretty severe symptoms. And the treatment that he received was extremely intensive behavioral therapy, which is the standard of treatment with this. Sometimes on the order of 40 hours a week. Everybody in the family gets trained. It becomes a part of how you interact with the person. And I told you that at the time, he wasn't putting a lot of words together. Well, he's, 25 now, and never shuts up. Just does not. Granted, nobody in my family is really good at shutting up, but in particular. And, if you met him right now, you probably wouldn't actually guess that he's autistic right off the bat. It might take you a little while to see that maybe he's got a little bit of social things, but he's doing pretty well. But that's really not the case for most children with autism. Because what happens is, if you are smart, if you have mild autism, then you respond to behavioral therapy. And you can learn the behavioral things, the social skills that happen automatically for a lot of people. You can actually compensate using intelligence. Well, what if your autism has this complication of intellectual disability? Then I can't really help you that much. But what if you come in, you've got autism, and I sequence your genome, and I say, you know what? You've got variation that fits with synaptic signaling. And you've got a SHANK3 version of autism. maybe now I can use my future magic genetic technology that doesn't yet exist but will, and I can help this child. And I just want to invite you to remember that autism itself is a very clinically defined disorder. This is a figure from Autism Speaks. It actually uses the slightly older definition of autism when language impairment was considered one of the main domains. And you realize that it just overlaps with everything, just like all the genes overlap with everything. Of course they do, because the way we assign the genes to diseases is our arbitrary way that we assign diseases altogether, right? You meet criteria, you get labeled with a particular type of diagnosis, right? And what I really want to start thinking about is this idea that, actually we'll come back to this, is this idea that, autism is not that big bucket at the beginning that I told you about, that said maybe keep that in your mind, I'm going to try to break that impression of it, but really represents this really diverse, broad category of disorders of human development. Maybe think about the idea that there's a lot of moving parts in the human brain. To make a human brain is very complicated. And sometimes things just go a little bit wrong because of the natural processes of mutation. And, suddenly, when I have just a little bit wrong, I get a child who can walk, whose bowels work fine, who can see, who can hear. Sometimes they can talk, but they just lose something else, something at the top, that social cognitive function or that higher order intellectual ability, depending on the type of autism you have. Kind of seems like that starts to fit with the idea that we're putting together here. All right, we've talked about genetics this whole time, and that only gets us to 60% of this pie. What else is going on? What else is causing autism? We're using genetic clues to tell us about what's going on in the brain, but that's, of course, not going to be the whole picture. Right? We have early childhood, like you think about things like early childhood infections. Maybe having particular types of infections when you're really little impairs your brain development. Maybe, I don't know. Environmental exposures. We know there are environmental exposures that increase your risk for autism. If you take a drug called Depakote when you're pregnant, you have about an 8-fold increase in risk of autism. So people who have seizures who take Depakote, we actually try to find other seizure medications that'll work for them before they decide to get pregnant, right? There's, I was talking to, this is something that I've done a little bit of research on myself, is there's an association between taking antidepressants in pregnancy and autism risk, right? So slight increase in risk, go from 1 in 110 to 2 in 110 risk. If someone comes to me and says, I'm really horribly depressed and anxious, I'm on this SSRI, but I just got pregnant, does that mean I should take the Musvin SSRI? Probably not. I certainly wouldn't, I would really think very carefully about it. But maybe understanding where those risk factors come into play with the biology will actually tell us a little bit more about the biology of what's going on in autism, right? So study serotonin cell development and its contribution to autism. Or study the effects of Depakote and study the effects of what that does to early brain development, and that'll tell us that, because that kind of mimics an autism phenotype that comes purely from genetics. And this idea of gene-environment interactions. So if you were to say, Sean, give me your best guess about what the rest of this pie is going to be, I would say that a big chunk of it, probably like another quarter of this, is probably going to come from your background genetic variation giving you a certain amount of risk that interacts with environment differently. Meaning, maybe you have Depakote exposure or SSRI exposure in utero, but you have very low genetic liability. You don't end up developing autism. But you have certain risk factors in your genome that interact with your environmental exposure. And suddenly, what wouldn't have caused autism in someone else causes autism in you. So environment itself plays with genome, back and forth, loop-de-loop-de-loop, all the way around. And so that probably is a good chunk of this pie. And hopefully, as we move forward, we can start building the rest of this out, understanding where the somatic mutations fit in, where the common variation fits in, where the environmental exposure fits in. And this is this almost kind of brute force method of trying to understand what is a very complicated disorder. So, big takeaways tonight. This idea that autism represents a very diverse category of disorders in human brain development. And what we call autism is really a clinical diagnosis. By that, I mean we have observations of behavior and what clinicians tell us. And understanding the basic biology of autism-associated genes and gene networks that we put together from autism-associated genes might actually help us understand the particular deficits in the autistic brain. Or brains would be the more accurate way to say this. And maybe that has something to do in the future with being able to develop personalized therapies, right? We hear a lot about precision medicine. This could be one of those categories that fit into it. So now, the idea, right, like we've talked about this, and I brought it back, I think about my idea when I said, oh, I want to understand autism, I want to understand what's going on with my brother, and I want to see if I can help him or help other families with this. And I started off in this kind of cognitive psychology side, right? That's where I started. And I realized very quickly, I was like, well, this is a little arbitrary. Maybe it's not truly arbitrary, it's functional, right? It's this, I can help this kid who behaves like this, I use behavioral therapy, and it works for some kids, and that's great. And as I started to study more, I got more interested in the biology of the brain, right? What's going on, cells communicating with each other, genomes, this whole thing is absolutely fascinating, right? Beautiful, complicated. And what you start realizing after years of looking into this is that, autism really represents this broad category. Like the study of autism is more actually the study of human brain development than it is the study of a particular disease. So if I were to go through and look at all 2,000 of those genes and write 2,000 PhD dissertations where I do really careful biochemical functional characterizations of these things and go through and do a really thorough job, like take Shank3, and put it in mice, put it in flies, check it in humans, do all sorts of things, write a PhD thesis, and do that for all 2,000. I think what would end up happening is you get a really, really good idea of what makes up a normal neuron and a normal brain. So what does this mean about what autism really is? Like, does it just represent natural variation in the human population? Right? You have this idea of we carry the same amount of mutations, but the types of mutations you have are different. Right? You carry more damaging mutations. You carry more rare copy number variants. So these kids just got unlucky where their mutations were, and that just changes how they end up developing. And this idea that autism maybe is part of evolution, right? This idea that this is one of those ideas that's tossed out a lot. The same things that it takes to make that complex brain machinery, to make a human a higher order thinker, have complex social interactions. Well, suddenly, when I build those things, now I have the potential for those things to be broken. So autism is kind of a uniquely human disease in that way. Or this common variation in autism, abstract thinking, pulling away from the concrete right in front of you, those types of abilities. Maybe autism is those taken to a far extreme, right? Too much of a good thing is a bad thing. So however you want to philosophically approach this, what we can realize is it's not one disorder, one disease. This clinical definition is useful functionally, right? I'm studying to be both medical doctor and a researcher, so my goal is to functionally help people that are in front of me, but also work on research that helps pull further in the biological characterization. So having functional definitions and scientific definitions are kind of two different domains. So however you want to think about it, there's a lot that goes on that tells us about the human brain when we think about autism. So with that, I just really, really want to thank my mentors who have taught me so much. I stand very, very much on the shoulder of giants, and I did not list so many people, but I have to talk at least about two. I mentioned Dr. Christopher Walsh, who's here. He's the reason that I came to Harvard, and he's the reason when I came to interview here, and I said, oh, I don't know, and he's the one who convinced me to come here, so I have to thank him for that. And maybe one day we'll be able to do a PhD with him. And my mentor when I was an undergrad at WashU in St. Louis and was Joe Doherty. And he's the one who worked a little bit on Self-6 and that SSRI project and taught me a lot about what it means to be a decent scientist. And also, both of them are kind, caring individuals. So lots of other thank yous for, in terms of where Science in the News gets funding. I've really enjoyed getting involved. It's my first time involved with the process. And hopefully I'll get to answer some questions after this too. But thank you so much for your attention. So with that, questions, please.
Okay, so where do some of these markers, mutations, etc., figure into things like high-functioning autism, Asperger's, etc.? Because everybody knows somebody that functions perfectly fine, but you go, there for the grace of God goes autism.
Yeah. Yeah, so is there a difference in the types of mutations for high-functioning autism and low-functioning autism? Right, so there's some talk in the field of one of the problems of trying to sequence a bunch of people and compare autism and neurotypical is that the bucket is so big and so diverse that you wash out a lot of your signal, right? A lot of the ways that you determine the difference between two groups, you compare signal to noise ratios, right? And if you deal with such a diverse group, you're going to wash out all the signal. So what if I actually take a more specific group of autism, only look at high-functioning autism. Maybe I cut off a threshold somewhere. maybe that actually yields some answers. And I would guess that there probably would be some changes. They have tried to do this with not a lot of success because, and it's really a problem of the fact that each one of these genes has a very small contribution in the overall picture of autism. So statistically, it means you need huge sample sizes to find things. And we're really only now getting to a point. But that is actually the idea of what those autism syndromes and finding those single gene disorders was based on, right? I'm going to look at a very particular cluster of autism, and oh look, there's something that explains that. So my guess is yes. Go ahead.
I have a couple of questions, but I'm just going to try to pick one.
Sure.
Has there been any research, to your knowledge, about the autism protein mutations, so the mutation specific to autism that have been discovered thus far, at the synaptic level? Has there been any research showing if certain proteins would be like inhibitory or cause like an excitatory postsynaptic potential, and if that were the case, would those be more related to schizophrenia type of overlap versus maybe an intellectual disability overlap? And we looked into like potentials.
That is a brilliant question. The question is, has there been anyone who's looked at the particular types of changes and mutations, put them in model systems, compared and looked at what they actually do to neurons, and how that fit into different diseases that the gene might overlap in terms of causing? And the answer is yes. Actually, that's really what, so there's two types of geneticists in the world. The ones that make gene lists and the ones that follow up on gene lists and do those types of experiments. So I would like to think that I actually fall into that second category, even though I gave you a whole talk about that first category, except for the end. So most of the work that I've done has been in not quite so specific as electrophysiology of the neurons, but there has been research. So I'm trying to think about a particular gene. So there was one that was on the synapse. It was like CANTB2. So it's a particular channel that's on the synapse that's important for ion flow that changes how the synapse behaves. Right. So people will look at that particular gene and do electrophysiology, where they actually measure the electricity at the neuron. And they can show that the different mutations in those genes actually changes the synapse itself and changes the signals that come out from one neuron to another.
If it was synaptic related and we go through synaptic pruning in age. Would those, is it possible that they could go away over time if it was like something that was, if the protein mutation was merely only at the synaptic level? Is it possible that it could, if you had it when you were younger, that you think it would disappear when you get older? The question is, if you imagine a protein that's in the synapse that's associated with autism, and then imagine as someone gets older, synapses get pruned and synapses get reorganized, so maybe you get the kind of this waning of autistic symptoms as you get older. The answer is probably not, because that piece in the synapse is actually going to be important for whatever synapse gets remodeled or formed. But there are genes that are on that who's who's list that are only expressed in development, not expressed in adults. Right? Or there are genes there that are expressed kind of later in like early childhood and not in development. So like you can look at the biology and say, well, this gene is disrupted in utero, but it's not even expressed in post-development, but I know that if you have a mutation here, you're more likely to develop autism. So maybe my intervention for this is some sort of gene therapy in utero, right? So where you catch a mutation. Now, of course, much of what I'm talking about here is really speculative, right? This is a field that's not in its infancy, but it is barely walking and talking right now. So, all the way in the back.
Yeah. There are many studies about the correlation between autism and mirror neurons, and most of them are in UCLA with young phonics actually who discovered mirror neurons.
Yeah.
So, are you looking for some genes for both mirror neurons that broke your empathy autism?
So the question is that there's been a lot of talk of things called mirror neurons and mirror neurons associated with autism. And are we looking for genes that are associated with the function of mirror neurons and autism, that kind of interface? So let me start with what a mirror neuron is for someone who, for people who haven't heard of them. Well, imagine have you ever had a feeling when you look at a TV screen and you see someone do some crazy gymnastics, and you know you can't do those gymnastics, but you're like, I could kind of do those gymnastics, right? Like this idea of you kind of feel what you're seeing. You see someone smile, you become a little happy. You see someone cry, you become sad. and this idea of empathy, of looking at someone, and this kind of like critical human behavior. And it's thought that this behavior of empathy is mediated by a group of cells called mirror neurons that fire, so right now you're looking at me, and I reach down and I grab my phone and I pick it up. This carries particular motor neurons in my brain to do that. But by watching me do that, some of your motor neurons will fire, even though you're not reaching for your phone and picking it up. So there's the idea of a mirror neuron. So you're really not going to like my answer because I'm really not a fan of mirror neurons. I think that they are a concept that has some truth, and they certainly exist in primates. But their role, even in normal human behavior, is more than controversial. And they're sometimes thought of as like overblown in their popularity. Again, this is something that's hugely controversial, and this is just my take on it as a very young, in-training scientist. But if you could find the functional genes that are important to make a neuron a mirror neuron, once you characterize what exactly a mirror neuron is, perhaps there might be an overlap between those and autism-associated genes, and then you might be able to see that some autisms are associated with a dysfunction in mirror neurons. But it's actually pretty far outside my area, so that's my guess. All right, so we'll take one more question, and then well, I'm happy to stay as long and talk to people, because one thing that makes scientists really happy is talking about science. So, yeah, right in the middle.
Yeah, so because you want to test a gene, you sort of need to knock it out in some sort of models. We're mentioning rodents and flies and worms. But how is the behavioral translation? How accurate is it to humans?
I'm so glad you asked this question. Yes. The question is, in order to determine the effects of particular autism-associated genes, what you use model organisms like flies and worms and rodents. But how do the behavioral changes in a rodent or a worm or a fly translate to human disease? Because autism is a human disease. And I used to work with mice, right? And I broke self-6 in mice, or worked on mice that had self-6 broken in them. And you know, what you realize very quickly is there's really no such thing as an autistic mouse, right? You have mice that behave differently than other mice, and you have flies that behave differently than other flies. But what you're doing when you're testing a knockout of a particular model is you're not creating an autistic mouse, you're creating a model of a mammalian brain that has a particular protein broken. And you're kind of counting on the fact that there are some similarities in the circuitry and some similarities in the brain development. There's actually a decent amount of them. And you're just using that to learn about what is it that this protein actually does in neurons? What is it that this protein actually does? So maybe there's no such thing as an autistic mouse, but I can break certain proteins in mice and show that they lose social behaviors. So somewhat analogous, right? So there are tests where I can test how much a mouse wants to hang out with other mice. Normal mice want to hang out with other mice. These mice tend to want that less, right? Or mice that show a lot of repetitive behaviors, that groom themselves a lot until the point they're kind of like whisking their fur off. So these types of circuits exist in early mammalian brains, and even earlier brains than that. But really, you're right. There's not a clean translation. That being said, animal models are actually a really critical way that you understand these. We have lots of tools for animal models. We have lots of standard assays. But the whole picture, you have to end up combining insights from animal models, from specific cell models, from tissues, from induced pluripotent stem cells and stem cell neuron models from human patients, and then consider the clinical picture of human patients who have that mutation. So you're trying to put all these pieces together to come up with an overall idea of what's happening, which is one of the challenges, right? You don't do research on humans, you just rely on the fact that sometimes a kid is going to walk in with a particular mutation. So that's the answer to that one. With that, thank you again so much. It's been a real pleasure. I'd love to talk to anyone who wants to talk to me. And I hope you come to future Science in the News events and lectures. There's information over there.